when I was preparing this talk, what I decided to do was to look at some recent headlines that came out, I looked 2 days ago to see what was out there, just to get an idea of what's kind of going on. So this is what I found. Genes drive our coffee habits, study finds. And this is a study that potentially found some genes that could affect how much coffee we drink. How about diets personalized to our genetic makeup might be more effective? That's also in the news recently. And then my favorite was, you could be genetically predisposed to be single, right? So basically what happened is that about 10 years ago, they had a huge development in technology for analyzing DNA, and because of that, with some very large-scale efforts, they sequenced the human genome. So for once, they had a map of the genome. They also identified all the common variation in the genome. So these are like some papers, kind of the seminal papers from the major studies at the time. It really ushered in an era of kind of new research, and many people came into the field, including myself. So I've been in the field about 10 years, and during that time, there's been a tremendous transformation in all areas of genetics, including understanding of genetics disease, cancer, population genetics, precision medicine, et cetera, okay? So just so we start from the very beginning, okay? This is basically human genetics 101, okay? So mothers and fathers, they make children, right? And in addition to making the children, they actually also pass on genetic information to the child. So each of us has 2 copies of each of our chromosomes, and when we have a child, randomly, one of them gets passed on to the child. So for example, in this case, this father passed on this green chromosome to the child, but if we had another kid, equally likely could have passed on this one. So children are about, contain half of the DNA, exactly half of the mother and the father, and the parents pass on half of what the DNA they have onto the child. So in addition to passing on chromosomes, another thing that's passed on is a disease risk. So if your mother has a disease, you're much more likely to also have the disease, okay? And the reason why that is because there's something called risk factors, and we'll talk about this in much more detail. Risk factors are genetic variants that increase your chance of having the disease. So if your mother has the disease, part of the reason why she has the disease is likely she has many genetic risk factors in her DNA, and since she passes on half of her DNA to you, you're going to get a lot of those risk factors too, which will also increase your chance of having the disease. So that's kind of the mechanism behind why, not just disease, but traits in general are passed on. So the things that we're interested today to talk about are the following questions. So first of all, how genetic is a trait or a disease? That's one thing we want to answer. And then the second thing is, how does genetics influence a trait? Like, what's the mechanism? And then the final thing is that with this new technology to measure our DNA, is it worth it for you to actually go in and get your genetics tested to see what you have? Okay. So the first question is, how genetic is a trait? And the way that this is really answered is by looking at twins, okay? Is there any twins here? No, okay, they're rare. But those people who are twins are very useful. And there's actually 2 kinds of twins, okay? There's maternal twins and paternal twins, okay? So here's some examples. So these 2 are maternal twins. Okay, we've probably seen them when they were younger, actually, for sure. Paternal twins are Arnold Schwarzenegger and Danny DeVito. And unrelated individuals, here's an example of a couple unrelated individuals. And the key thing is that maternal twins share 100% of their DNA. That's why they're called identical twins. Actually, they're called identical twins because they look identical, but their DNA is identical. Paternal twins share about 50% of their DNA. Not exactly. Paternal twins, for all intents and purposes, are just like siblings. They're born at the same time, okay? And what the interesting thing about, if you look at traits, you can look between maternal and paternal twins and get some insights on how genetic something is. So what we compute is something called concordance, okay? So basically, a concordance is the following quantity. So if one twin has a disease, what's the probability of the other twin having the disease? Okay, that's called concordance. And for example, let's do some examples. So for example, diabetes, okay? if you are an identical twin and your twin has diabetes, you have a 34% chance of having diabetes, okay? But if you're a paternal twin and your twin has diabetes, you have a 16% chance of having diabetes, okay? Similarly, if like you're just sitting next to some guy who you're totally unrelated to, your chance of having diabetes will be 8%, which is just the population average, okay? So the difference between 34% and 16% is exactly how much that other half of the genome, the DNA being shared, causes the disease. So using these two quantities in large numbers of twins, we can actually measure how genetic a disease is. For example, diabetes is relatively genetic. Schizophrenia is also highly genetic. If you have a twin that's schizophrenic, your chances of being schizophrenic is like 40%. Identical paternal twin, it's 5%, and just in the population, it's like 1%. Tay-Sachs, for example, if you have an identical twin, it's basically 100% chance you're going to have the disease. If you're sibling, then there's like a 50% chance, and if you're unrelated, then there's less than a 1% chance. So that's basically how genetic a trait is. So we can look at like, okay, so these diseases, like, you know, what's the genetic basis of these diseases? Okay, so there's actually can be split up into really two types of disease. And so the first type of disease I want to talk about is what are called Mendelian diseases. So these diseases are very rare, okay? So, and typically they're characterized with a single gene that has a very large effect. And these were actually discovered in the late 80s using family studies. I'll talk about that in a second. But examples, we've heard of these. These are like Tay-Sachs as an example, cystic fibrosis as an example, Riley-Day as an example. So these are kind of like diseases where, you know, for example, Tay-Sachs is essentially completely due to this one gene, like hex A. If you have two copies of that gene with a variance, you're going to have the disease. Cystic fibrosis is also with the CFTR gene, et cetera, okay? So what is the kind of mechanism for the genetics of these Mendelian diseases? So this is actually how it works. So for example, Tay-Sachs, each individual, all of us, have one of three statuses for the disease. So we're either unaffected, okay? We are affected, or we are carriers. Okay? Actually, the carriers themselves are unaffected, but I'm just using that term. So the way that the way that the disease works is that if you have two parents that are carriers, then they, with a 25% chance, this kid will inherit both chromosomes that have the mutation, and then if you have two copies of the mutation, then you'll be affected. Okay? So with a 25% chance, if you have two carriers, you'll have an affected kid. 50% chance, these two scenarios, a kid will be a carrier, and with a 25% chance, the kid will actually be like completely unaffected. Okay, so this is like an example, like Tay-Sachs is like this. That's why you might hear, you know, carrier, not carrier. If like you and your partner are both carriers and you have risk, if you're a carrier but your partner's not a carrier, then there's no risk. So genetic testing for these kinds of diseases are highly accurate and informative, widely used, and the way that all these genes were discovered was basically people, like geneticists took families where they had some affected kids and some unaffected kids and kind of looked at the differences in their DNA, and they were able to find all these genes. So this is actually for many rare these diseases, they know the gene, they can do a gene test. Okay, so a question that kind of comes up is, why do, like the Jewish populations in particular, why are they more common? Like, why do they more commonly have these diseases? For example, Tay-Sachs outside of the Jewish population, it almost doesn't happen. Okay, so the reason actually has to do with kind of the Jewish population, particularly the Ashkenazi Jewish population, has a very unique history. That essentially, about 700 years ago, there was what's called a founder effect. So essentially, you can think of that 700 years ago, the Jewish population was only 350 people, and since that time, it expanded to whatever the 10 million that we have now, and that everybody essentially is related through these individuals, and that basically made many of these extremely rare disease variants that were prevalent 700 years ago, very rare, in the Jewish population because only 350 people kind of propagated. What happened is that any one that they had became really, really common. So that's it's kind of the reasoning why, and there's a recent paper actually that showed this. Okay, so that's those rare diseases, we've heard of them. So what about the more common diseases? So these are called complex diseases, and so examples are diabetes, depression, kind of heart disease. So in this case, these are all common diseases. Many of these diseases affect more than 10% of the population. Actually, these diseases will end up killing most people. Most people die of one of these diseases. And the reason why they're complex diseases is that there's many genes that are involved in these diseases. So unlike these Mendelian ones where there's only one gene, here you have many genes with very small effects. And so the search for these things, actually like complex diseases using families in the 90s completely failed. And for a long time, actually, there was almost no progress in genetics until relatively recently. Okay, so actually, here's actually how complex diseases work. Okay, you can think about diabetes. So these are all genes that are implicated in diabetes, and there's actually hundreds more. So in each of these genes, there's some genetic variant where if you have it, it'll either increase or decrease your risk of diabetes. Similarly, there's a bunch of environmental factors that affect diabetes, diet, exercise, pollution, also luck. If you're just lucky or not, you might get diabetes or not. So here's actually, luck is a big factor, okay? So here's a hypothetical example, okay? Let's say you have, all but these two variants, okay? And then let's say for your diet is of cake, exercise, yoga only, you live in LA for pollution, and you have no luck, okay? So you can think of the way that we think of this kind of complex disease is that each of these factors, whether genetic variants as well as environmental variants, they give you a score. Okay, so you get maybe you get a score for all the variants that you have, and maybe they sum up to, in this example, you get a genetic score of like 63, and so cake, whatever, is not good. Yoga, it's okay, but not great. LA, pollution's gotten better, it's good. Luck, also not good. So your environmental total is 43, and then your total score is the sum of those. And let's say in this case, if your score is higher than 100, then you have the disease. If your score is lower than 100, then you won't have the disease. So you can see there's an interplay. For example, if you have better genetics and you have a lower genetic score, you can eat more cake and you'll be fine, okay? If you have a very high genetic score, then what will happen is that even if you run marathons and stuff like that, you're going to be very close to having the disease. So that's the basic idea, okay? So, but like I mentioned, in the 90s, we didn't know what these variants are because they're very small. So what happened, actually I want to mention here that there's a few exceptions to this rule. So there's this BRCA gene in breast cancer that actually has an extremely large effect. Similarly, there's this ApoE4 gene in Alzheimer's that also has extremely large effect. And also autism doesn't really fit the model. It fits the model, but not this description. It's like you have many variants with large effects. But those are pretty much among the common diseases, the only exceptions to that kind of genes with small effect rule. Okay, so how do they find these genetic factors? So this is actually something that really started relatively recently. So here's the idea. The idea for finding these genetic factors is what they did was they just took lots of people. Okay, for example, they might have taken 2,000 people that have diabetes and 2,000 people that don't have diabetes. Okay, the people who have the diabetes are called cases. Those are individuals that carry the disease. The people that don't have diabetes are called controls. That's kind of the background population. And what they did was they looked in each gene to see if there was any genetic variance between those two groups. And they collected people so that they expected them to be about the same age, about the same ethnicity, so that everything else is the same except for their disease status. And so the idea is that if they see some kind of genetic differences between these two groups, then those genetic differences might be actually causing the disease. So let's do an example. So this is an example. So these are, these this is kind of what the DNA looks like. So it's A, C's, G's, and T's. It's this is a small example. The actual sequence in each of us is 3 billion. And so let's say this is like the DNA of individuals that have the disease, and this is the DNA of individuals that don't have the disease. And then, so you can see that actually all the black positions, they're the same in both groups. Okay, that's because about 99.9% of human DNA is the same in the entire population. So it's only these like red positions which are different. Okay, and they're called single nucleotide polymorphisms. Okay, but for short, they're just called SNPs. So if you want to make it sound like you know when you're talking about human genetics, just say SNPs, and people will be impressed. Okay. And so if you look really carefully here, you can see that one of these variants is associated, okay? So if anyone has really good eyesight, you can kind of see. And so it actually turns out to be this one here, okay? So in this one, almost all of the guys that have the disease, the cases, they have a G, while almost all the guys that don't have the disease have a T. So that's what we mean by an associated variant. And so that variant might be responsible for the disease. And so with some more effort, you can actually validate that. So that's the idea behind this association analysis. And so by 2007, they had the technology to do this, and then they applied this in a very large scale. So this is just gives you an idea of like what's going on in the field. So this is actually the number of publications on these kind of genome-wide association from 2005 to 2013. Okay, so what you notice is that in 2005, there's almost nothing. Okay, basically they didn't know anything, and by 2013, they've actually had now 2,000 different studies on these association studies, and so they found a huge amount from almost knowing no genes that are involved in complex disease. Let's give you an idea how this changed. So this is actually in 2009. Each of these vertical bars is a human chromosome, and each of the colored dots is a location in the DNA that's associated with a common disease. And so this is give you an idea. In 2009, this is how much they discovered using these genome-wide association studies. This is what they discovered in 2010. So there's many more dots. This is 2011. Or actually 2012, and then this is 2013. So you can see that there's more and more and more that's known from all these studies. So this is actually huge progress. There's almost, when I got into the field in 2003, all the geneticists were really depressed because they spent most of their careers without finding anything. So yeah, I came at a good time, I guess, right? So this is what we found. So now the question is, okay. We talked a lot about DNA, so your genes can tell you something. So there's some signals that are extremely loud, okay? These are the rare genes, okay? They have very large effects. If you're unlucky enough to have one of them, then they can tell you something. And then also, but for complex diseases, the signals are very small, okay? And for complex diseases, there are also other variables like behavior, environment. So the question is, should you listen? Should you get tested? Is there a point to do genetic testing? Okay, and so for rare disease, okay, this is now pretty standard care. There's many successful scenarios. So there's child rare disease diagnosis. There's for parental carrier testing, it's actually integrated very, it's very successful, and there's actually a lot of clinics that actually do this. It's actually even part of standard care in a lot of places. Those are actually very successful, and similarly, this BRCA, which is a breast cancer risk gene, it also has an extremely strong effect, and so that's actually widely performed as well. And those are kind of successful scenarios. The information from those are useful. Sometimes they give better disease diagnosis for sick kids, and then they can find what treatment would work better. And so for this, I would say that this is typically done through either a doctor or a genetic counselor. It's kind of it's pretty mainstream medicine now, and there's also some of the challenges are cost for paying this, and so there's some nonprofit organizations that actually pay for the cost, but they're also very much connected to the medical community. So I would say that this is actually kind of a very successful area. And so the question is, what about your genetic destiny for other things, okay? And so I want to actually revisit the coffee, okay? And so what I did was I looked at the coffee, and this article, and I found the paper, okay? And I decided, okay, let's look at what will this, they discovered, what will this actually tell us? So they actually looked at, this was a serious study. I thought it was not a serious study because I didn't really think that the Coffee and Caffeine Genetics Consortium was like a real thing, but it actually is. And they collected 120,000 people and basically measured their coffee intake and looked at their genetics. And so what they found was this. These are 8 genes that they found, and if you have the variant in the gene, this is how much it increases, this decreases or increases the amount of coffee you drink by cups per day, okay? And this is statistically significant because they have 120,000 people. So the thing is that if you look at this, if you have this one, that means that you're gonna drink 0.04 less cups per day, okay? Actually, in fact, if you have all of the genetic variants that will increase your coffee consumption, okay, it's going to increase your coffee consumption by 1/4 of a cup, okay? If you have all of the genetic variants that decrease your coffee consumption, it's going to decrease your coffee consumption by 1/3 of a cup, okay? But if you ask in this room, if you measure our coffee consumption, it's going to vary by a lot. Some people drink 3 cups, some people drink no cups. So the actual genetics here is very much drowned out by other factors, non-genetic factors. So that's actually very typical for these complex traits. So that's actually, for complex traits, very often the effects are very small. Okay, so where are we now? Okay, so in terms of everything is genetic. Okay, the twin study pretty much showed that every trait has a very large genetic basis. Okay, we've taken every twin we can find. I worked with a guy that literally collected every twin in Minnesota and asked them like every possible question and measured them in every way. So we know almost everything is genetic. Also, for rare diseases, we know we have many genes now. Genetic testing is clinical practice. And actually, even an example, there's actually some treatments that are available. Actually, in the State of the Union yesterday, the president mentioned that there's a new cystic fibrosis drug that just came out that's like a wonder drug that's also based on some genetics, which is great news. On the other hand, they discovered the gene 25 years ago. So in general, medical research is like a slow and steady field. For common disease, so we've discovered a huge amount. The field's moving very quickly. But what we found is that the effects of genetics are extremely small, and so the genetic testing is kind of in very early stages. Okay, so finally, what I want to do is now give you the tools to basically to determine whether when you read one of these articles, you know that it's true or not, okay? And so this is the basic mechanism. This is the basic rules, okay? So if it's a common disease or common trait, and there's less than, for it to be true, you need to have more than 2,000 individuals, and also it needs to report a very small effect, and it needs to do what's called a replication, which means that they have to also essentially do the study twice, okay? And otherwise, it's not true. So if we look at the coffee, okay, so they looked at 120,000 individuals. Their biggest effect is 0.14 cups of coffee per day. That's really small. And they did a replication. So we can conclude that probably what they reported is true. So you actually have this variant. You will drink slightly less coffee than if you did otherwise, okay? What about this one? The genetically predisposed to be single. So in this case, they looked at 600 individuals, and the results said that your 20 is being single. I would say that's a common trait, okay? And they found that you're 20% more likely to be single if you have this variant, okay? So clearly, it's not true, okay? Finally, the genetics of food. Actually, this is really interesting because also 138 individuals. 40 milligrams a day change in sodium intake. So clearly, in some ways, it's not true. But then when I read the actual article, they actually didn't. The actual article wasn't. All the newspaper articles said that if you have this personalized diet, you lose weight. But the actual article said that if you tell people that you're genetically really bad at processing sodium, what happens is that a lot of them stop eating sodium, okay? So that's what the article said. So if you tell people, people will believe the genetic results, even if they really don't have an effect, okay? So that's actually what they found. Okay, so that's it. Thanks. So this is my lab, and what I actually work on is gene by environment. So I talked about how we have common variants affecting a trait and the environment, but actually what I'm interested in is how some variants are only active in some environments. Like for example, if you eat a high-fat food, then some variants might be active, but if you eat really healthy, then they won't be. So that's called gene by environments. That's the main thing I work on. So thanks.